هاي بونسوار أول شي للكل وميرسي إلك ملاك ولين for contacting me. أكيد I'm a proud graduate. نبلش أول شي I'm a very proud graduate من Lebanese University, so that's why one of the reasons اللي I accepted this. وأكيد هدفنا كلنا نساعد، لأن بالنهاية كلنا at some point كان بده مين يساعدنا يعني to guide us. As Malak mentioned, I'm a first-year PhD student in a co-direction in between Lebanese University and the University of Montpellier. I'm under the supervision of Florence Camas and Derek Julien here in the lab of Institut de Recherche en Cancérologie de Montpellier, and Raghida Abu Merali from the Lebanese University. And today I will give a very brief introduction about my PhD project, because I obviously cannot present everything, which is entitled The Transcriptional Functions of the Lysine methyl-transferase sub-420H2 in the Progression and Metastasis of Prostate Cancer. Okay, so again, a brief introduction of myself. I did all of my studies in the Lebanese University, my master's one, my master's two, the genomics and health program. I did my internship. I was lucky that I did my M2 internship here in France in the same lab that I'm doing my PhD in. And the title of my internship was to characterize the role of heterochromatin protein 1 in response to stress, and the stress was the paracetamol overdose. And now I'm pursuing my first year of PhD. So just like a small introduction about my project, to start with, epigenetic modifications, they are important regulators of genome functions and that are impaired in cancer. Just because not everyone is familiar with the term epigenetics, epigenetics is briefly everything that affects the genome externally. So the genome, as we all know, the organization, it passes through several stages of compaction, beads on string, and all of these things that we know. In order to fit in the nucleus, and these different compaction levels, they obviously affect the levels of the transcription, like at the transcription level and further on the protein level. So these epigenetic modifications that occur at the level of the genome, they occur mainly at two main sites. We can either have DNA methylation, or we can have histone modifications on histones, mainly H3 and H4. Other histones can be affected, but it's mainly H3 and H4. These histone modifications, they include either phosphorylation, methylation, acetylation, ubiquitinylation, ribosylation, and all of these. And the newer ones, they are sumorylation. The different histone modifications on different histones, and specifically either on lysines and arginines, they have, if I can say, a specific outcome of that. Like, for example, H3K9 trimethylation is, you can say it's a mark of the heterochromatin, which is like reducing the transcriptional activity, whereas the acetylation is usually a mark of a euchromatin, which is the loose open chromatin, but it's not always the case. There are always exceptions, but generally methylation is correlated with heterochromatin, acetylation with euchromatin. So again, to start with the introduction of cancer, this normal homeostasis of the chromatin affects the state of these cells, and again their fate and their functionality. So any difference or like any change in these in this homeostasis or at the level of these modifications, it leads to a change in the fate of the cells, because they will eventually have to re-regulate themselves to cope or to adapt to the new changes. And most of the times they will eventually acquire what we call the cancer phenotypes, which is the cancer aggressiveness, metastasis, and all of these stuff, including genetic alterations. So the dynamic of chromatin, or like the source of these histone modifications in DNA methylation, is what we call readers. We have readers, writers, and erasers. So briefly, writers are the enzymes that put on the methyl or the acetyl group on the N-terminal tail of the histone. This I want to mention specifically because histones are modified on their N-terminal tail, which is the only part of the histone that is actually protruding to the outside to be exposed to these enzymes that eventually will put or will write like the epigenetic mark, which can be either methylation, acetylation, etc. We have the erasers that remove these marks, like demethylases, deacetylases, and writers usually have the methyl-transferases, acetyl-transferases, so they transfer acetyl. And we have the readers, which are actually proteins eventually that recognize the marks that are added by the writers. So eventually, like if we're speaking about lysine 20 at the histone H4, we have specific readers that either recognize the dimethylated form of this lysine, the trimethylated form, the monomethylated form, and eventually specific readers will bind to their specific mark. And so any mutation or aberrant expression of these epigenetic enzymes, it's gonna eventually misregulate all of this dynamics of the chromatin, leading to cancer. So this leads me to my family of protein of interest, which are SUB420H enzymes. They are methyl-transferases, so they are writers. They are the ones that I'm going to start from the end. They induce the formation of the di and trimethylation of the lysine 20 on histone H4, and they are the only known histone methyltransferases for histone H4 at the lysine 20 till now. So when we speak about histone H4 methylation of the lysine 20, we are mainly talking about SUB420H1 and H2. And in order to form these di and trimethylated forms, they use the monomethylated form as a substrate for this methylation reaction. And the monomethylated form is formed by also an epigenetic enzyme, which is called SET8, that uses the usual non-methylated form of lysine 20 to form the monomethylated. And then the monomethylated is used by SUB420H1 and H2 in order to form the di and the trimethylation forms. So I'm mainly interested again in these enzymes because the lysine 20 methylation on histone H4 is not very well studied and elaborated, especially in the cases of cancer. We mainly see articles concerning lysine 9, 27, lysine 4, 37 on histone H3, but very few on histone H4. And as I said before, the different methylation marks, they are recognized by different reader proteins that bind in order to form various functions, eventually cellular functions after. So SUB420H1 and H2, they have a structure of an N domain, a set domain, which is the catalytic part of the protein that I mean by catalytic part, which is the part that is able to form or to catalyze the formation of the di and the trimethylation form of the lysine 20, a post-set domain, and a C-terminal domain. Also, SUB420H1 is also known as KMT5B, and SUB420H2 is also known as KMT5C. So these SUB420Hs, they have different expression profiles during development, where we can see that SUB420H1 is ubiquitously expressed during embryogenesis and in the adult tissue, whereas SUB420H2, which is specifically my targeted protein of interest in my PhD, is restricted to adult tissues. And also the knockout of SUB420H1 in mice it leads to lethality, and if not lethality, severe developmental defects in the newborn littermates. Whereas in the knockout of SUF420H2, the mice were completely viable, no developmental deformities whatsoever. And so again, just to emphasize the idea more, that the alterations in SUF420H1 and H2, the mutations and the aberrant expression, leads to neurodevelopmental disorders and cancer. So just a brief idea of what are the different cellular functions of SUF420H. They are involved in chromosome compaction and segregation by their interaction with cohesion complexes. They can be also involved in DNA replication origin by recruiting the ORCs, which is the origin of replication complexes. They are involved in DNA repair by recruiting a very important protein in the DNA repair pathway, which is 53BP1. Yet their transcriptional role is still weakly studied in comparison to also the other epigenetic enzymes. Like, for example, I'm going to say like SUF39H1, it catalyzes, and H2, it catalyzes the trimethylation of lysine 9 and histone H3. And so it's like the transcriptional roles of these epigenetic enzymes is very well elaborated and studied in articles, yet again for SUF420H it's still poorly understood, specifically in the cases of cancer. So a small introduction about prostate cancer, since I addressed it in my project, and eventually it's the model I'm working on. It's prostate cancer is one of the most common types of cancer and like highly recurrent among men. At first, prostate cancer, if detected early on, it can be treated at some point with androgen deprivation therapy, because prostate cancer tends to be dependent on androgen, the hormone androgen, and because usually in prostate cancers, early stages, we have an increased expression of the androgen receptor, so more receptors, more binding of androgen to the receptor, and all of this pathway, which if I can say, makes the prostate cancer even more aggressive. So if detected early on, yes, androgen therapy can be a way, yet some patients, they will suffer from relapse after passing through the metastatic stages, leading to a castration, what we call a castration-resistant type of prostate cancer, and even more aggressively, the neuroendocrine subtype, which is what we call NEPC. This NEPC is the most resistant and the most lethal form of prostate cancer. So here we reach a state of like a blocked path, because they become resistant to all sorts of chemotherapy and other androgen deprivation therapies. And so this progression through the stages of prostate cancer is associated with the sequential accumulation of genetic and epigenetic alterations, leading to the formation of this castration resistance. So, oops, I didn't show the slide, but it's okay, I'm going to say it orally. From previous, the data from like the lab previously before I come, and from other sources, we have shown that this castration-resistant type of cancer in this metastatic form is often associated with increased expression of SUF420H2, but not of H1, and eventually we are having an increase in the level of the trimethylation form. And so we wanted to try to figure out why is SUF420H2 having an increased expression, and what is the correlation with that. And so here comes my thesis project, which is to identify the pro-tumoral functions of H2 in the context of prostate cancer, and eventually on the longer term to improve a therapeutic strategy based on the targeting of SUF420H2 functions. I will be mainly, like in the title I said, the transcriptional role, and for that I will be showing you a few examples of the RNA sequencing data analysis that I have been doing, because we have RNA sequencing data from in vitro, from the in vitro cell line of the, and the in vivo from xenografts in mice. And so eventually, after the analysis of the RNA-seq data, I will be validating and the interesting genes by RT-qPCR and immunoblots, and eventually trying to get to know more about the underlying mechanisms and how they are affecting prostate cancer, etc. So again, previous data from the lab showed that the loss of SUB420H2, but not of H1, it impairs prostate tumor growth in vivo, where for new people and young scientists, we take the cells that are in culture, we prepare them in a certain way with like a solution called Culture-X. We do the transplant into the mice, yet this is a xenograft, but not an orthotopic xenograft. And so we like xenograft the cells into the mice, and we wait for tumor growth, and then we measure the tumor growth. So wild-type cells, they were able to grow tumors. Knockout of SUB420H1 cells, they were able also to grow tumors. SUB420H2 knockout, no, they couldn't form tumors, or if yes, they are very, very, very small compared to the H1. Yet the double knockout, which is also interesting, is they are restoring the capacity of growing tumors again. So we went further from that in order to do the in vivo RNA sequencing. So this is just an experimental strategy for new people in this domain to just try to get along of what we are doing. So we extract the tumors, we extract the RNA, we send it for sequencing, and we analyze. So just a few slides on my analysis of the RNA sequencing data. Here I'm just showing you that in the cases of double knockout versus the wild type, in comparison to the SUB-H2 knockout versus the wild type, we see 401 genes that are upregulated and that are specific to SUB420H2. It can be, I know, difficult and like, how can I say, not so easy to understand for everyone, but it's just the analysis of the RNA-seq. It's eventually it is like that. So that's why I am saying specific to H2, because I am taking the genes that are upregulated in the case of H2 knockout versus the wild type. So you are comparing to the presence of both enzymes. And so what are these 401 and what are they involved in? Mainly they are involved in transcription and DNA repair, which is actually promising. And if I look at the genes that are independent of H1, which here the purpose was just to minimize the list of H2 dependent genes even more, to try to find the genes that are, or like if I can say in other words, that can truly explain the observed phenotype in the xenografts. I found 79 genes. And from these 79, they are also involved in sodium transport and angiogenesis. Concerning the downregulated genes, the same conditions of comparison, I see 555 genes. And among these 555 specific downregulated genes, you can see genes involved in lipid metabolism, which is this like in the path of me reading articles along with my analysis. It's like at the moment is greatly like if I can say focused on and elaborated the role of all of these metabolism pathways and everything in the progression and the metastasis of cancer, which is something new yet something interesting. I know that not everyone likes biochemistry and metabolism, neither did I at first, but trust me, it is interesting to see how lipids, glucose, all of these metabolic pathways, they truly affect prostate, especially that, again, as I said earlier, prostate is eventually is a cancer that at first is dependent on androgen. So even in that, because the androgen receptor pathway and different metabolic pathways, they are interconnected at some point. So seeing them downregulated was really interesting, along with immunity and innate and inflammatory response genes like HKDC1, apoptosis, Wnt signaling pathway, and the proto-oncogene, it's called MET. Here it's the same condition for the H2 specific genes that are independent of H1. I can find 195 genes that are also involved in lipid metabolism, immunity, fatty acid metabolism, and all of these stuff. So the goal later, like right now, is to validate these RNA sequencing data by RT-qPCR. Sorry. So what I basically did is I extracted the RNA, and you do the reverse transcription again to get back to the complementary DNA of your messenger mRNA that you amplify. And then I perform the usual RT-qPCR by adding the gene primers of the targeted genes and the buffer. And you do the qPCR reaction with the machine, and then you analyze the data. So concerning MET, the proto-oncogene I told you about earlier, it's showing, yes, that it is specifically related to SUFFER20H2 and that it is a specific gene for H2. Yet we still don't know if it is a direct target or not. And so it positively regulates the expression of the proto-oncogene MET in my prostate cancer cells. So we see in the wild type cells we have, which is what we compare on, the knockout of SUFFER20H2, we have downregulation of MET. In H1 knockout, we have an upregulation of MET. In the double knockout, it's downregulated, but not as the same as the H2 knockout. And these two last bars, they show the cell lines overexpressing H2 and the rescue of H2 in the SUFFER20H2 knockout cells. And this is also just the validation by Western blot for MET. And we can see that in the H2 knockout, we have indeed downregulation of MET, but the effect is not clearly seen at the protein level as much as like at the transcriptional level, which even enhances and boosts the theory or the hypothesis that I started on. The SUFFER20H have a transcriptional function or transcriptional regulation function. And so here again, I also tested and I analyzed the qPCR for the gene expression of HKDC1. HKDC1, I will not go further into details because it is a very complex hexokinase. It is involved in the glucose metabolism simply by two words, is that it traps glucose inside the tumor cell. So the tumor cells will have, if I can say, more fuel to use and to grow more, metastasize. And it is involved in the inflammatory response by enhancing tumor evasion. So it's also suspected to be to have a pro-tumoral function in cancer and to be specific to SUFFER20H2. So in conclusion, prostate cancer is shown to have an increased SUFFER20H2 expression, and this is correlated with the metastatic stages of the disease and the neuroendocrine phenotypes, which are again the most resistant and lethal forms. The SUFFER20H2 knockout cells, they are associated with specific gene expression changes, which may or may not explain the observed phenotype in the xenografts, and we may find other genes we still don't know. And the rescue of the SUFFER20H2 and SUFFER20H2 knockout cells, it partially increases the levels of MET and HKDC1 in these cell lines. So indicating that at least till now, MET and HKDC1 are yes specific specific targets of SUF420H2. We still don't know again if they are direct targets or not. So if SUF420H2 binds to their promoter or not, this is for a future perspective, obviously. But yeah, by that I will try to end my talk, not to elaborate even more. Again, thank you for the invitation. If you have any questions, please don't hesitate to ask. And yeah, that would be basically it. Yeah.
Hello, Hiba. Very nice presentation.
Thank you.
I had a few questions actually. يمكن أكتر شيء أكتر out of curiosity due to the vastly different domain that you're working on. You mentioned at the beginning, slide 6 specifically, إنه كان في differences or there are different forms of methylation, either mono, di, or tri. My question is, and you said that the different readers read these differentially methylated histones, right?
Yes.
قد بتفرق I don't know if we should speak in Arabic or English, but to
Yeah.
what extent does it? Yeah, to what extent does it matter, or how vast is the difference between the mono, di, or tri-methylated forms that are being read? Is it like, for example, an amplified effect if a protein or a histone is tri-methylated, or is it just completely doing something completely different based on what the readers are reading?
Okay. To start with your question, Ali, is that I just where is it? Yeah. Okay. Just like again to go back, the dimethylated form of histone of lysine 20 on histone H4 is the most abundant form of like the most abundant form that we see in the genome in general. The trimethylation mark and the monomethylation mark, so they represent 10% each, so the dimethylation is 80%. So going back to your question, yes, the different reader bindings to their specific methylation marks, they induce specific functions according to the reader protein that binds to the, like for example, 53BP1 is involved in repair pathways, and so it generally binds to the monomethylated form and the dye. But for example, HP1, which I talked about that I studied in my M2 internship project, it's mainly correlated with the trimethylation form to induce to maintain, if I can say, the heterochromatin state, because it is by its name heterochromatin protein 1, so it is just to maintain and to fix the heterochromatin in this closed, very compacted form to restrict transcription. So he مش بس at the extent of how much or how much not they bind, but at the level of what is that binds to this specific form, and eventually the function, because it's a specific reader, so it will do a specific function in the end.
That's extremely interesting, actually. I have another question regarding a slide that you showed. I don't think I have the number of the slide here, but you mentioned that you're going to be working, or you have worked with both in vitro and in vivo models. I'm curious to what extent, I mean, obviously the in vitro models are never going to be perfect, but regarding the in vivo models, maybe this raises more questions. To what extent are the effects that you observe within mice correlational to what you would expect occurring in humans? And are there any major limitations when it comes to these effects?
Yes, okay. I will start from your question in the opposite way. It's not me who did the xenografts in mice, but I have a few info. Again, to start from the end, one of the limitations which I said, I think, during my talk is that these are xenograft models. So these cells, they are not actually grafted in the site of prostate. But what we are planning to do further on in the future is to try to do orthotopic xenograft, which means you xenograft in the prostate itself. And we are trying to develop a new cell line other than the one I'm working with, where you can follow the metastasis of the cell line by using a fluorescent protein. So you see properly if this cell of its specific genotype, like H1 knockout, H2 knockout, has the ability or not to metastasize and to form eventually tumors in the environment of the prostate itself. So at the level of limitation, yes, it is a xenograft. It is not the best model to start with, but at least it's better than just showing or like seeing in vitro, like the cells in vitro, because I didn't show it also here because I don't have the time, Ali, but all of these knockout cell lines, they proliferate equally the same as the wild type cells. So there is no difference at the level of proliferation, at least in vitro. So having at least this early on model of xenograft is like a further step. But again, it's not the most perfect. So yeah. So obviously, if it's not xenografted in the area of the prostate itself, mainly there will be also other changes from like the environment, the surrounding, I don't know, receptors or like other proteins that can be recruited to the zone of the prostate, and so yeah, it's really complex. But as I can answer till now is that. So yeah.
Nice. I just had one last question regarding the Western blot that you showed at the end or towards the end. So you showed that the H1 knockouts, well, more or less lead to no difference between that compared to the wild type. But I mean, this immediately presents the question, why did he decide to go for a double knockout in this case? I'm not sure if I missed it in the talk, whether a double knockout leads to any other differences. But for me, at least just looking at this, purely just looking at this, I would say the effects would be strictly from the H1 knockout, independent whether you have the H2 knockout, excuse me, the other way around. This is strictly, let me rephrase it. The effect that we see within the double knockout is strictly what we see because of the H2 being knocked out and not the H1, if that makes sense. Is that accurate, or is that not very?
Yes and no, because the value of the log fold change in from the RNA sequencing data is not as low in the double knockout as compared to the H2, which this arises the question if you compare it with HKDC. Sorry, I'm moving on to another just to get back to your question, is that here in the case of the hexokinase, in the double knockout, we see again an overexpression or like an upregulation of HKDC, which this implies that HKDC1 is like restricted, like its function, transcriptional level is affected, if I can say till now, not 100% sure, but solely by H2. Whereas MET, yes, I cannot deny that H1 may have an effect on MET, but it's also following the path of H2 to some extent. And so this also raises the question of maybe it's either H2 is acting on like a repressor or an activator of MET that will, so here in this case, the knockout, yeah, it depends if it's activating a repressor or repressing an activator. So, or if MET is a direct target of H2. So if H2 binds to the promoter of MET or not. So yes, it can be an effect of H1, you're right. But at first, what is important is just the validation of the RNA-seq, which was successful, at least if I can say. These are like the results of the MET and the qPCR and the Western blot. I got them 2 weeks ago. So we are still in the process of trying to understand the effect of H2, H1, or both of them on these at least 2 effector genes. But yeah, so they obviously have different patterns of expression or like different dependence on H2, H1, and so yeah.
Thank you.
You're welcome. No more questions? Don't be shy. I know you students are still like in the beginning, but don't be shy. We were like you, but don't be shy. Thank, thank you. Thank, thank you. See you. Bye.